Number 1. Define genetics and its importance in fishery. The benefits may include improved growth, resistance to disease, or robustness in diverse farming environments. Genetics can help to improve fishery management. It is very important in fishery. In terms of breeding program, efficient breeding program will be crucial to this development, not only to reach the production goal but also to reduce production costs, improve disease resistance, improve utilizations of feed resources and improving product quality. The history of animals breeding show that the principle of selections have been applied for thousands of years, long before the theoretical background was developed with considerable success. The main purpose of a breeding program is to develop animals which more efficiently utilize available feed, land, and water resources. It is very probable that by pursuing this goal, we will also select animals which are better adapted to the captive environment, as they must produce well under this condition. Number 2, what is the chromosomal and chemical basis of heredity? According to this theory, chromosomal is a gene that are the units of heredity and are found in the chromosomes. Chromosomal theory of inheritance came to existence long after Mendelian genetics, and diazoxyribonic acid is the chemical basis of heredity in organism. DNA is a presence inside the nucleus. It contains genetics information and instructions for protein synthesis. Number 3. Explain the concept of population genetics. Population genetics is the branch of genetics that conduct the study of the genetics compositions of populations. It's combined genetics, evolutions, natural selection, selective breeding, statistics, and mathematics. It's make computers and mathematical model and conduct field research to test the model. Population genetics spend their time doing two things: describing the genetic structures of populations or theorizing about the evolutionary forces that affect populations. Number 4. What the importance of selection in fish stock management? Selectivity operates both at a local scale as in the direct interactions of individual fish with the fishing gear, contact selections, and at a stock-wide scale, population selection, as evidenced by the differential rates of fishing mortality at age that are the generally observed in stock assessment result. Number 5. Differentiate hybrid from inbreeds and plant a good crossbreeding program. Hybridization is the process of crossing genetically different individuals to produce offspring, whereas inbreeding is the crossing of two closely related parents, who share very similar alleles. Inbreeding is more likely to cause genetic abnormalities, unlike hybridizations. Number 6. Identify biotologies in fish genetics and determine how they could help increase productivity. By biotechnology are chromosomes manipulations and hormonal treatment which can be used to produce triploid, tetraploid, haploid, geogenetics, and androgenetics fish. This results in the production of individuals and lineage of sterile monosex or highly endogamic fish. Culture fish are being improved for a multitude of traits, including growth traits, feed conservation, efficiency, disease resistance, tolerance of low water quality, cold tolerance, body shape, dress out, percentage, carcass quality, fish quality, fertility and reproductions, and harvestability. Number 7. Differentiate mitosis from meiosis. Mitosis is a process of nuclear divisions in eukaryotic cell that occurs when a parent cell divides to produce two identical daughter cells. Mitosis is conventionally divided into five stages known as prophase, prometap- prometapase, metaphase, anaphase, and telophase. Meiosis is a process where a single cell divides twice to produce four cells containing half to original amount of genetic information. These cells are our sex cell, sperm in male, eggs in female. During meiosis, one cell divides twice to form four daughter cells. Number 8. Differentiate DNR from RNA. DNR is a model-stranded molecules while RNA is a single-stranded molecules. DNA is stable under alkaline conditions, while RNA is not stable. DNA and RNA base pairing is slightly different since DNA uses the bases adenine, thymine, cytosine, and guanine. RNA use adenine, uracil, cytosine, and guanine. Number 9. Discuss how and why DNA replicates. DNA replication is the process by which a double-stranded DNA molecules is copied to produce two identical DNA molecules. Replication occurs in three major steps: the opening of the double helix and separations of the DNA strand, the primering of the template strand, and the assembly of the new DNA segments. During separations, the two strands of the DNA double helix uncoil at the specific location called the origin. DNA replicates because whenever a cell divides, the two new daughter cells must copy the same genetic information or DNA as the parents of cell. Number 10. Explain the importance of transcription and translation. The goal of transcription is to make RNA copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide protein or protein's subunit. Eukaryotic transcript need to go through some processing step before translation into protein. Translations enables effective communication between people around the world. It is a carrier for the transmission of knowledge, a protector of cultural heritage, and essential to the development of a global economy. Highly skilled translator NK. The purpose of transcription is to make RNA copies of individual genes that the cell can use in the biochemistry. The purpose of translation is the synthesized proteins, which are used for millions of cellular functions. Discuss how protein amino acids are synthesized. During translations, ribosomal subunits assemble together like a sandwich on the strand of mRNA, where they proceed to attract tRNA molecules tethered to amino acid circle. A long chains of amino acid emerge as the ribosome decodes the mRNA sequence into a polypeptide or a new protein. Number 12. Explain the principle of YY male technology and the process on how to produce YY males. YY males produced by converting female into female population. Number 13. Explain the different kinds of artificial selection method: mass selections, family selections, and multi-trait selections. Mass selection. The term individual selection and mass selections are often used interchangeably, and they refers to selection solely based on the individual phenotype. This is a well-known and widely used method of selection in animal breeding, and for several aquaculture species can be used on characteristics that can be measured in the candidate's animal itself, as weight gain being most efficient in characteristic of high heritability. Family selection refers to a selection method in which family groups are ranked according to the mean performance of each family and whole families are served of discarded. The difficulty of the individual marking of the animals, which is still a fact in quotations, led to the recurrent use of this method in fish farming, with the constant impediment of the emergence of the family effect. Multi-trait selection. This is the opposite of the previous method. The selection is based on the deviations of each animal from the average of your family. There is two choice of animals of all families. Then the method requires identification of the families. This may be achieved by maintaining them in separate tank cage, hapas, or any other means of containment without unnecessarily tagging the fish. The full sea group rear in a prelicated hapas or any other form of containment fall into this category. Under such circumstances, selections between families will be misleading from a genetic viewpoint because of the confounding between genetics, means, and common environmental effect. Its replacements are chosen to that every family contributes the same number of individuals to the next generation. Choose one female and one male from each family. The effective population size is twice the actual. Number 14. Advantage of crossbreeding. Crossbreeding is used to produce superior fish, grow out production fish, while selection is used to create superior brood fish. One of the most common breeding programs in aquaculture is the production of sex-reverse brood stock to produce monosex population for grow out.